Ever wondered what makes us unique, each with our own distinct characteristics and traits? The secret lies in the infinite world of DNA sequencing. Imagine a vast, intricate library, brimming with volumes of information. Each book represents a single organism, and every page within it is a unique chapter of genetic information. This is the world of DNA sequencing, the process of determining the precise order of nucleotides within a DNA molecule. First, a small sample of DNA is extracted, usually from blood or saliva. This DNA extraction process is like the librarian carefully selecting a book from the library's shelves. This DNA is then broken down into smaller, more manageable fragments. Consider these fragments as individual chapters of the book. Next, these fragments undergo a process known as amplification. In this stage, millions of copies of the DNA fragments are made, ensuring there's plenty of material to work with. Imagine a photocopier, duplicating the chapters so that they can be studied in detail. Following amplification, the real detective work begins. A machine called a sequencer reads each DNA fragment, determining the exact order of the 4 nucleotides: adenine, thymine, cytosine, and guanine. It's like reading through each page, word by word, to understand the story. Upon sequencing, the fragments are reassembled to form a complete DNA sequence. This step is akin to putting the pages back together to understand the full story of the book. Finally, bioinformaticians interpret the DNA sequence. They identify genes, variations, and other valuable information that can tell us about an organism's characteristics and genetic health. It's like a literary analyst interpreting the book's narrative and themes. So what did we uncover today? We delved into the fascinating world of DNA sequencing, exploring how a simple sample of DNA can be transformed into a detailed genetic blueprint. We learned about the steps involved, from DNA extraction and fragmentation to amplification, sequencing, and interpretation. DNA sequencing, in essence, is a remarkable journey of discovery. It allows us to understand the genetic makeup of an organism, opening doors to new possibilities in fields like medicine, agriculture, and environmental science. It's like reading the story of life itself, written in the language of our genes. Remember, each DNA sequence is a unique story, waiting to be read. And who knows? Perhaps one day you might be the one to read and understand these stories, unlocking the secrets of life itself.